Good evening, ladies and gentlemen. Welcome to the Monash Biomedical Imaging Centre at Monash University. My name's Lawan Ismail, the Executive Director of the ICT for Life Sciences Forum. It's terrific to have so many of you here this evening. It's my pleasure now to invite the Director of the Monash Biomedical Imaging, Professor Gary Egan, to introduce this evening's presenter. Gary?
Okay, good evening everybody, and welcome here to MBI. It's an absolute delight this evening to be able to introduce David Van Essen. David is the Alumni Endowed Professor of Neurobiology in the Department of Anatomy and Neurobiology at the Washington University in St. Louis. He's internationally known for his research on the structure, function, connectivity and development of the cerebral cortex in humans and non-human primates. And that goes through decades of incredibly significant work that David and his colleagues have undertaken. He's been the leader of the development of many powerful methods of computerised brain mapping, both in non-human primates in his earlier work and more recently in the human. He also has been a pioneer in the neuroinformatics tools that are needed to be able to do that sort of research, and the development of some very early tools that I remember playing around with, Carrot, for example, back in the 90s, and more recently some terrific neuroinformatics tools. David's also the principal investigator of the Human Connectome project, which is a NIH-funded project which has been now running for a number of years, and we'll hear more about that tonight, which is in particular, I think, setting a benchmark for the sharing of data across the neuroscience research community. My understanding is it was actually part of the actual conception of that Human Connectome project to make the data available as soon as absolutely possible to the research community. I know that's actually happening in real time because some of the members of my group have got some abstracts going into conferences using data from the HCP, and then we've started collecting similar data here at MBI and adding to that. David has served in many leadership positions, including, as I mentioned, chair of the Anatomy and Neurobiology Department at Washington University. He was the founding chair of the Organisation for Human Brain Mapping in 1996. He was a counsellor of that organisation from 1992 to 2002. He was the secretary and president of the Society for Neuroscience in the United States, president from 2006 to 2007, secretary from 2002 to 2004. Absolutely eminent positions within the field of neuroscience. David's a fellow of the American Association for the Advancement of Science and has received the Raven Lifetime Achievement Award from the St. Louis Academy of Sciences and the Krieg Cortical Discoverer Award from the Cahal Club. So please join me in welcoming David to give his lecture here this evening.
Thank you, Gary, for that kind introduction and for the opportunity to talk to you about some of my scientific passions over the recent years. I'd like to start with some numbers. I love numbers about the brain, and let's remind you that our brain has approximately almost 90 billion neurons. The dominant structure, of course, is the cerebral cortex, but it has only a minority of the neuronal count, about 20%. That's because it's chock-a-block full of literally hundreds of trillions of synapses to communicate information. And then the long-distance traffic goes by way of almost 100,000 miles of cabling in the underlying white matter. The cerebellum is the pint-sized player, but it actually has 80% of the neurons, even though it's only 10% of the brain mass. And then we're kept alive, and hopefully for most of us well most of the time, by virtue of the subcortical structures, loosely called the rest of the brain, which has fewer than 1% of your total neuronal count, but is hugely important for keeping us awake, hungry, or satiated, and so on. So our overarching challenge is to figure out what's going on within this incredibly complicated network of connectivity that achieves our healthy human brain function. Analogies with computers are often made and are apt to look at the similarities and differences. So a computer maybe larger or smaller than our brain, but basically it has computational elements that operate on incredibly fast speeds compared to our neuronal poking along at hundreds of spikes per second as the maximum rate of communication. When we think about understanding computers, we have to be mindful of what's the underlying hardware, what's the operating system that makes it function, and what are the apps that do good things. For our own brains, we need to understand the wiring of the brain just as the wiring of the computer is incredibly important. We want to decipher the signals at macroscopic and microscopic levels, and ultimately we want to understand how this mediates our uniquely human behaviors. And we'll see we're making headway on that, but it's an incredibly challenging and long-distance project to work through all these issues. Our focus today is on connectomes. I think everyone in the room is familiar with genomes, which are incredibly precise delineations of every base pair up to our 3 billion DNA sequences. For the connectome, we have to put it in major quotes right from the outset because we aspire for comprehensiveness, but the reality is for the human brain, we can only realistically aspire for examining long-distance connectivity down to the resolution of the imaging elements, the millimeter to 2 millimeter voxels that we'll be talking more about. There is a wonderful world of microconnectomes that can examine and reconstruct every neuron, glial cell, dendrite, axon, and synapse, but literally only to sub-millimeter scales to date. And then there's an increasingly important world of what I like to call the mesoconnectome that uses tracer methods in animal models to trace local and long-distance connections down to, often down to micron level resolution. But we'll focus for the rest of the day on the macroconnectome of the human brain. And our agenda for the remaining part of this talk is to first put more context for thinking about the cerebral cortex in both animal models and in humans, and then dive into the Human Connectome Project proper. And let's see if this one, that works a little better. Good. So the cerebral cortex is the dominant structure not only of our own brains, but of mammals in general. Our brains are 3 times larger than that of our closest living relative, the chimpanzee, which in turn is 3 times larger than that of the macaque monkey, an intensively studied animal model, which in turn is 100 times larger than the intensively studied mouse model. And the marmoset is the smallest of the laboratory non-human primate models that the Rosa group and others are making into another important model system. So the cortical convolutions vary enormously from humans down to the lissencephalic mice, and that raises the issue of cortical cartography. How do we deal with these convolutions? This became apparent to me when I dived into this domain several decades ago, and at that time it was not possible to turn the job over to computers. I'd literally developed a method using pencil and tracing paper to make a flat map of a piece of monkey cortex. When I moved to my independent lab at Caltech, John Mansell and I were able to make a flat map of an entire monkey hemisphere and to paint on it essentially the geographic landscape, the gyral and sulcal patterns, and we could also paint on it the functional landscape, the equivalent of the political landscape of an Earth map, by using information from different modalities and different sources. So identifying the distinct cortical areas that define the visual cortex and other domains draws on information about cortical function, architecture, connectivity, and topographic maps of visual fields or other body domains. There were at the time and still are a small number of poster children, essentially the areas like V1 and MT that are exquisitely well defined by multiple modalities, and they're very popular for study for that and other reasons. But the reality is that most of these other areas were and still are hard to delineate. The boundaries between them are subtle by whatever criteria one uses, and there's noise and bias bias and individual variability that adds a confound, and so we see to this day discordant results from different analyses. So cortical parcellation is really important, really hard, and I'll come back to that again and again. The best parcellated mammalian cerebral cortex is now the mouse. It's cut to the head of the chase thanks to some elegant work by Andreas Burkhalter's group, who have identified 40 cortical areas in what is not completely the final word, but I think is the closest to ground truth that we have to date. For the macaque monkey, as I said, it remains a challenging problem, and we can take a macaque cortical surface and let a computer reconstruct it and inflate it and paint onto it a parcellation that has several times more areas than in the mouse. So it is comprehensive, but everyone would acknowledge that this is not ground truth, that there is ongoing debates about the existence or extent of many of these areas. For the human cortex, we have even more severe challenges that we'll be delving into, but as of a couple of years ago, my colleagues and I published a summary map of the 52 cortical areas that at that time were well defined and mapped onto cortical surfaces. Altogether, those occupy approximately a third of the cerebral cortex. There are Brodmann maps and other parcellation pieces for the intervening regions, but basically the way I've looked at it is if you fill in the gaps, you could predict that there's going to be somewhere between 150 to 200 human cortical areas, and where we are in delineating them I'll come back to. But now let me turn our attention for a few moments to the basics of cortical connectivity, again starting with the macaque as a model and going back to ancient history. When I got into this business, there were a limited number of anatomical tracing methods, and this seemed kind of complicated at the time, but little did I realize that the advent of increasingly powerful retrograde and anterograde tracer techniques would allow John Mansell and me to identify a more complex collection of areas and connections, and also to delineate a hierarchical organization based on the laminar patterns of forward and feedback connections, as Kathy Rockland had shown and we followed up on. Dan Fellman and I took this to another level, and those of you who saw my tie have seen this subway chart version of macaque connectivity for just the visual areas. So we could again use this hierarchical scheme to identify many levels of processing and literally hundreds of pathways. That seemed incredibly complex, but little did we know what was in store. We were also in a position to take the same several hundred pathways and to turn them into a connectivity matrix, essentially a parcellated connectome by our current parlance, and to identify the presence or absence of inputs from any one visual area to any other in this 32 by 32 matrix. But this was a binary connectivity matrix, and it was frustrating because we knew at the time that some areas were powerfully connected and others only weakly so, but there was precious little quantitative data despite the obvious need for it. Jim Lewis and my lab pursued the quantitation of cortical connectivity, and this is one example of a quantitative plot that we were able to generate, but it was very tedious, painfully slow work, and we were not able to push that as far as one would like to see ideally before Jim escaped with his thesis. So a decade later, the field had to wait until Henry Kennedy's lab put a decade of their own laboratory's intensive efforts into the systematic quantitative analysis of macaque cortical connectivity. So basically, they did the same kind of thing that Jim Lewis had done, inject area 7A, identify and count quantitatively every retrogradely labeled cell, and then we worked with them to paint these long distance and nearby cortical connections onto a surface-based atlas. So you can see there's a lot of connections, and if you look quantitatively at the strength of pathways, you see this logarithmic plot shows that there are some pathways that are very strong, some that are incredibly weak, 5 orders of magnitude more sparse, and the majority of pathways are intermediate, but every injected area showed this same kind of log normal distribution of connection strength. So it's not just there or not. If one wants to get a handle on connectomes, one has to be mindful of these quantitative realities. Putting the variations in strength aside, on average for the 29 areas that the Markov et al. Studied, there was an average of 55 cortical inputs from just each hemisphere. And when one tallies the total from that extensive analysis, there emerges a 29 by 91 quantitative connectivity matrix with over 1,600 pathways identified. And if you stretch that to ipsilateral and contralateral and subcortical connections, we should be thinking of non-human and human primate brains as having 10,000 or more pathways that we want to get a handle on. So that's a daunting order. Turning our attention briefly to the mouse again, this parcellation that Andreas Burkhalter worked out, they also carried out quantitative analyses of connectivity and confirmed that there are many orders of magnitude of connection strength and lots of pathways, and in a real tour de force analysis carried out by the Allen Brain Institute, the effort that Alan Jones has spearheaded. There are in the mouse connectome, again, thousands of pathways ranging in connection strength. So as we now move to the human cortex, this is our grounding and what we should be prepared to countenance and try to explore. So when we look at human cortex, the first thing that I think it's important to concentrate on is the nature and degree of variability in cerebral cortical convolutions. So we can take here 4 individual cerebral hemispheres, just the right hemisphere, in healthy adult individuals, and look at the variability of the folding pattern in the what we call the mid-thickness surface, or in inflated views on the right of the same individuals with maps of what we call sulcal depth. The darker it is, the deeper it is in the buried cortex. And so you can see it's highly variable across individuals. For example, up here in prefrontal cortex, there's great variability in the folding pattern from one individual to the next. Ditto for occipitotemporal cortex. And remarkably, this degree of variability occurs even in identical twins. So as it happens, here's one pair of identical twins, here's another, and look at the individual differences in the folding patterns. So there are genetic instructions, and indeed there is a quantifiable heritability to cortical folding patterns, but what strikes one in the face is that the variability dominates over the heritable components. Another critical thing to have in mind is that the cortical areas that we've talked about already are highly variable in their size, that is in their surface area from one individual to the next, and also in their folding, the relationship to the underlying folds. So that's been known for a long time. Here's a nice example of it from the Amundsen and Zillis efforts over the years. The red is V1, the green is V2, and you can see how variable they are in their location, and they vary in twofold or more in surface area. So this brings me to an intriguing aside, not central to our main story, but I think it helps to provide some context to think about why and how does the cerebral cortex get its folds? And indeed, why is the folding pattern so radically different from a dolphin to an elephant to the virtually lissencephalic manatee? What makes this pattern species specific and distinct for each individual within a species? Well, one starting set of observations is that cortical folding occurs relatively late, but during prenatal development in primates and including humans. So here is a preterm infant right at the beginning of the 3rd trimester, very lissencephalic, and during the next several months, the folding pattern gets well established, and at birth it's pretty close to but not identical to the adult size, even though the brain is only 1/3 of its full size. So my favorite scientific light bulb moment was when I was thinking one evening about how folding occurs, and in the narrow sense of visual cortex that I worked on, I knew that V1 and neighboring V2 are massively interconnected. That is the most powerful cortical pathway we know about. And it occurred to me that the gyrus that forms in between them in adult could arise if mechanical tension along the long distance pathways, say from V1 to V2, if those axons were under tension, then they would get pulled closer together and a gyrus would form in between them. And that idea, once initiated in the narrow context of V1 and V2, generalizes to all of cerebral cortex and generalizes to brain morphogenesis in general as an attractive but still unproven hypothesis. But in the context of human cerebral cortex, it has the attractive feature of accounting for consistency of folding, for instance, in the central sulcus and the Sylvian fissure in regions that are strongly connected by relatively large areas, and yet the variable patterns of folding may be accounted for by the balkanization of many little cortical areas that have differences in size and connectivity from one individual to the next. So we still don't know if it's true, but it's a way of thinking about the issues that now come to fore when we dive into consideration of human cerebral cortex and the human Connectome project. So I'm going to break this into multiple parts. I'll give you an overview of what the objectives of the project are in broadly writ and where some of the accomplishments we've made overall. Then I'll dive into some specific methodological advances and some of the kinds of discoveries we're starting to make, and then wrap it up in due course. So the first objective of the Human Connectome Project and the consortium centered at Washington University and University of Minnesota is to acquire data on multiple modalities about brain structure, function, and connectivity in healthy adults drawn from a population of twins and their non-twin siblings. We're using an improved scanner with a high gradient strength for better diffusion imaging and using advanced pulse sequences that I'll say a little bit about before. We get 4 hours of data from each of our volunteer subjects in 1 hour slots over 2-day periods, and we get multiple modalities that I'll be illustrating in detail. The quality of the data by our inspection and by multiple criteria and what we hear from others is exceptionally high for MRI standards. We are about 2/3 of the way through the data acquisition phase, about 800 scanned to date, and by a year from now we expect to complete our target number of 1,200 adults. We're also scanning 200 of these individuals who agree to fly up to Minnesota and be scanned at the 7 Tesla scanner in Camille Lugerbill's group there, and we're about 1/4 of the way through that effort. We're also gathering several hours worth of behavioral data to allow correlations and comparisons between connectivity, function, and behavior. We're getting data from 100 subjects using magnetoencephalography, and we've got blood samples that will plug into the genotyping pipeline at the very end of the project. We're not just acquiring the data, we're doing extensive early analysis using a set of pre-processing pipelines that turn out to be critically important for capitalizing on the quality of the incoming data. And as Gary alluded to, we've developed improved visualization tools. I'll show you the outputs from Connectome Workbench, and I'll show you some of the more advanced analyses that we've been busy carrying out. As Gary alluded to, a core part of our mission is to share the data, and we have that not in a warehouse, but in a well-organized database called Connectome DB, built on the XNAT platform that Dan Marcus's group had pioneered, and we've refined and extended to handle the multiple modalities that we have acquired. And we've already released data from 500 subjects last June, and more to come, obviously, in the future. We're getting excellent buy-in from the community. More than 2,500 investigators have signed on to our data use terms so they can easily get the data. We're approaching a petabyte worth of data sharing. About half of that comes from high-speed downloading from Connectome DB, and another half comes from what we call Connectome in a box that you can order the hard drives at cost. And then we're releasing extensively analyzed data. I'll illustrate some of that. We've already released an initial bolus of MEG data and another big bolus coming next month. And we're getting a lot of traction on our HCP website. We're also sharing the tools that we've spent a lot of effort in developing. So Connectome Workbench has already been downloaded over 2,000 times. Our pipelines that I touched upon have been made available as of last month. Many sites are using the acquisition protocols that I'll touch upon later that benefit data acquisition. And publications are now starting to stream out. Gary alluded to abstracts and action going on here and elsewhere. Lots of people are asking questions about how to capitalize on the data, and we're getting a lot of good feedback. And I can't say enough positive about a really fabulous group of investigators, more than 100 on the team, who've really worked arduously and passionately to make all of this happen. So what is in fact happening in terms of scientifically interesting data and results? Well, I'm going to give you some examples of what we are acquiring and how we're interpreting and analyzing the data, and we'll go through this whole laundry list in relatively short order, I hope. So the first thing is to get the best possible structural imaging data. So here on the left you see a high resolution 0.7 millimeter T1 weighted scan, a standard structural image, and you can see that the FreeSurfer algorithm for capturing the shape of the peel and white matter surfaces is doing a great job. We took Bruce Fischel's FreeSurfer and worked with him to make further refinements to it, and you can see the kind of surface rendering we can obtain for this. We're also acquiring T2 weighted images, and the combination of T1 and T2 weighted data affords us not just a better representation of brain structure, but access to another kind of modality that we call myelin maps. So the starting point is if you take the central sulcus here and look at the gray matter ribbon, a T1 weighted image is brighter in some regions and darker in others. If you take a T2 weighted image of the same region, there are also variations in image intensity within the gray matter ribbon, and it turns out that the ratio of those, the T1 to T2 ratio, is informative about myelin content. So Matt Glasser, who conceived and implemented this whole myelin mapping approach as a student in my lab, has colorized this slice to show regions of heavy myelination and lighter, sparser myelination. And we also compared this to architectonic subdivisions of the somatomotor domain and showed that these myelin map transitions correlate with anatomically definable architectonic boundaries that the Zillis lab and others have analyzed over the years. So this can be done not just for a patch, but for an entire hemisphere, as you see here. And it can be done to show these myelin maps for the left and right hemisphere, inflated and flat map views. And you can see that there's a systematic organization of heavily myelinated patches and strips, red and yellow in a systematic way. And also the lightly myelinated domains are informative as well, that most of them, we believe, are correlated with architectonic or functionally relevant subdivisions of cerebral cortex. Moving on to fMRI and diffusion imaging, those two modalities have benefited greatly from the advent of improved image acquisition methods, slice accelerated or multiband image acquisition pioneered by Camille Lugerbill's group at CMRR and also by the MGH group, complementary approaches. And the basic concept is that rather than acquiring data tediously one slice at a time that takes many seconds to get through the entire brain, one can excite using the RF pulse sequences many slices at a time and then reconstruct the scene of the crime using multi-channel head coils. We use a 32-channel head coil, and it, basically one can do the math and reconstruct with remarkable accuracy the origins of the MR signals. And for the fMRI in our Connectome pulse sequences, we can have a multi-band factor of 8 that gives us higher spatial resolution than typically is available and faster timing. 0.7 seconds is essentially the movie rate, and both of those are highly beneficial. For diffusion imaging, for technical reasons, the multi-band factor is only 3, but that's still a big gain that lets us get very high spatial resolution for diffusion imaging. So the good news is it gets higher quality data. A price one pays is there's a lot more data from the improvements in spatial and temporal resolution, and that brings us to another component of how we've approached this problem, which is to make best use of the data and focusing on the parts that are the components that are of highest interest. So for functional MRI and the domain that we care about is the gray matter of cerebral cortex and subcortical gray matter structures. And the other stuff, the white matter and CSF and non-brain tissue, is there in image volumes, but you don't need to hang on to that. And so we developed this approach that we call focusing on gray ordinates, gray matter domains in a data format that we call SIFTY, kind of like NIFTY and GIFTY that have become standards. We hope this will become an emerging standard that uses surface vertices to represent the cerebral cortex, uses good old voxels, but just a subset of gray matter voxels that are of particular interest for fMRI. And for the cerebellar cortex, we would like to capitalize on the fact that it is a sheet of tissue, and I'll come back to that later, but we cannot accurately reconstruct the cerebellar cortex in each individual because it's such a thin sheet. So we compromise and represent it as gray ordinate voxels for the time being. Another important aspect of dealing with the data is to do the best possible inter-subject alignment. And for cerebral cortex, the natural domain is the cortical sheet, and I'll come back in detail to the different ways of surface-based alignment that we've been exploring. But for the subcortical structures, one can do very well using nonlinear volume-based registration to align each individual to an atlas template. Another point that's conceptually and practically very important is that when one acquires fMRI data, it's noisy, and for improving signal to noise, one needs typically to do some kind of smoothing, but our general position is that smoothing should be done in gray ordinate space. That means respecting the cortical sheet for the cerebral cortex and representing respecting compartmental boundaries for subcortical structures. So we use this gray ordinate space to get the data-rich, what we call dense connectomes that constitute 30 gigabytes of data per subject and would be 3 times as large if we didn't use this efficient representation. I should note that the concept of gray ordinates is specific to the fMRI domain. For diffusion imaging that we'll come back to, the important domain is white matter, but we can extend the concept to white ordinates as the collection of voxels that represent the white matter of any individual. So coming back to fMRI, we get an hour's worth of data in 4 15-minute aliquots from each individual at this high spatial and temporal resolution that I'd mentioned before. I'm not going to go through the details of pre-processing of the volume data or the surface data, but just suffice it to say that Matt Glasser and others on our consortium spent a lot of effort into thinking very carefully about how to pre-process the data to maximize alignment, minimize any distortions, and also Steve Smith and colleagues have developed a denoising strategy that they call FIX, and it does a great job automatically removing the noise from an fMRI time series. So here is an exemplar time series from one individual subject to show you the fluctuations in the fMRI bold signal that is essentially an orchestra of fluctuations in space and time that is the input to the next level of fMRI data analysis. So if we take this kind of time series data, one of the important kinds of functional connectivity analyses is to do a correlation analysis, pairwise correlation of the fluctuations between any one gray ordinate and all of the others. That leads to this concept of a dense functional connectome that's 90,000 by 90,000, 32 gigabytes worth of data of just the gray matter. And then we can interrogate this functional connectivity map one row at a time. So at location gray ordinate vertex number 1, this is the functional connectivity map of high correlations nearby and at a distance, and the anti-correlations in other regions. And at another location 2, it's a very different functional connectivity map. So this is one level of representation that is highly informative, but we'll be wanting to process it further. But I want to take another sidebar moment or two to comment on where we are at and why we're glad that we're acquiring data at 7 Tesla as well as at 3 Tesla. So the starting point is a reminder that cortical thickness varies a lot. This is a group average of 210 subjects, and basically you can see that there are some regions where the cortex is 3 millimeters to 4 millimeters thick. On average it's about 2.5, and there's some places where it's quite thin. Conventional fMRI acquires voxels at 2.5 to 4 millimeters in size, and you can see it's the partial volume price is rather large in that conventional mode. For our 3 Tesla Connectome standard acquisition, we're on the low end of the spectrum, but for the 7T acquisition, we were able to dial down to 1.6 millimeters and still get excellent signal to noise and better spatial detail. So in a preliminary analysis, as these data have just come off hot off the press, when Matt Glasser and colleagues looked at the an estimate of what gain we're getting for the 7T data. One way of looking at it is to say on an average of 24 subjects, how many bold signal components are we getting? And basically it's almost twice as many for 7T as for 3T. Another measure is to look at the contrast to noise, and again it's an almost twofold difference. In practical terms, one can see that by eye by taking a seed region of the group average functional connectivity. This is again a dense connectome map with a seed and parietal cortex, and for the group average at 3 Tesla, one sees a pretty high quality dense connectivity map, but you can see at 7T it's much improved in terms of signal to noise. And when we do analyses such as looking at the gradients in functional connectivity that inform us about functionally relevant parcellations that I'll come back to, you can see the group average gradient map for 3T is not nearly as strong as that for 7T. So we're gaining substantially in this, but 7T is a bleeding edge technology, and we think we made a good choice in not putting all of our eggs in that basket, because it took a long time to get us to this stage. Okay, moving on to the next set of topics, dealing with individual variability. The core issue for cerebral cortex is improved intersubject alignment that leads into better parcellations that I'll come to. But first, for the intersubject alignment, the starting point is bearing in mind the high degree of individual variability, especially in many regions such as prefrontal and lateral temporal and parietal cortex. using shape features alone has until recently been the only game in town for aligning across individuals. And that works actually very well as Bruce Fischl's free surfer intersubject alignment method. It does a great job at aligning primary visual cortex. It does not do so well in other regions such as this MT complex or HOC5 architectonic area, where the variability across individuals just doesn't get aligned using shape features alone. So after a long effort led by Emma Robinson and Mark Jenkinson, we have a new game in town, an algorithm called multimodal surface matching. It's very sophisticated, over my head in terms of the technical details, but I appreciate that the outcome is excellent. It uses a discrete optimization method, and importantly, you can choose which features, shape features and functionally relevant features, and you can tune the parameters that constrain the registration. So as I'll show you, it compares favorably with other methods, and it's working great so far on the Connectome data sets that I'll be showing you. So one point is that using shape features alone, free surfer, though it does a pretty good job, has had a reality that is essentially over-aggressive. And if one aligns individuals with very different folding patterns, it will force aerial distortions that are larger than seem biologically natural. So this is a group average aerial distortion map that's achieved by standard free surfer alignment. When we take the multimodal surface matching and start using just the shape features alone, the same kind of constraint but with different implementation parameters, the average distortion is substantially lower with the MSM method. It's about more than twofold lower by multiple measures, and so that's good news for reducing unwanted distortions, and it actually improves the quality of functional alignment. And so we've used this shape method that we call MSM Sulc for the sulcal depth measure as what we are releasing publicly, but there's more to come with further improvements. So here, if we take this MSM shape-based method, the MSM Sulc, and look at the group average myelin map and the group average functional connectivity from an exemplar seed and parietal cortex, you see pretty good alignment across individuals, but one can do substantially better by using the resting state networks, the functional connectivity, as an additional constraint on this MSM-based registration. And so here you can see the same data set, but with an added booster shot of alignment. What was pretty blurry here is a sharper transition with the MSM functional constraint, ditto for another exemplar location, ditto for these others, that both the myelin map and the functional connectivity benefit from this improved method. However, while that is nice, in a way it's predicted because we use the resting state networks as part of the constraint, and so it should improve the alignment. But what about other modalities such as the task fMRI that we've also acquired? Does it improve the alignment of that modality as well? And as you can guess, I'm showing you because the answer is yes, it does help a lot. But to set the stage, I should point out that we acquire an hour's worth of task fMRI data from every individual, 7 different tasks. One of them is listening to stories versus doing arithmetic in the scanner. Another is a social cognition, determining whether this is random movement or hugging and kissing of little triangular icons. A working memory task is another, and 4 other tasks I won't show you. But to illustrate the flavor of what we get, this is again a group average task activation pattern for listening to stories versus just fixating the background fixation spot. So in the group average, what one can see is a pattern of yellow and red activations and blue and green deactivations in this individual or this population. And the working memory task shows you a complex network of activations with a very different spatial pattern. So these are two exemplar task contrasts with the starting MSM SOLC, the shape-based alignment that I alluded to before. When we take the same data and now using the resting state networks to improve the alignment, you can see that indeed the alignment is improved for the task activation, even though that was not part of the data that was constrained the registration. And this amounts quantitatively to a 15% improvement in alignment, which you might say, well, that's not so much, but we think it's actually a pretty big deal given the challenges of extracting high-quality fMRI data. So now with that under our belt, let's look at the parcellation problem, which I've touched upon before. I'll remind you of the importance of it. With dense connectivity data, it's just overwhelming the amount of data one gets in individuals and group averages. So people have long recognized the importance of dimensionality reduction, but using neurobiologically plausible subdivisions of the cortical sheet and the brain in general. So there's been a lot of progress outside the domain of the Human Connectome project in using resting state networks to parcellate the brain into functionally distinct networks. So Randy Spuchter's group has done this and has found resting state networks that are broadly dispersed. They are spatially non-contiguous in ways that are interesting about network organization of the cerebral cortex. There are other methods that enforce topological continuity or contiguity, which is natural given the way we think about cortical area parcellations, as I've touched upon them from classical approaches. So there are multiple methods that have been published recently, but there's still room for improvement. And there's also an issue that's very important to think about. Functional connectivity gives us information about regions that have similar fMRI time courses, but it's been known for a long time and now appreciated that it's really important to recognize that regions such as the somatosensory motor strip has functionally distinct compartments for the different body parts. The face region and the body and hand regions have very different functional signatures. So fMRI resting state analyses do not map cleanly onto architectonic areas of the type that I've described before. So that's motivated Matt Glasser for his thesis work in my lab to use a multimodal approach to cortical parcellation. So we have the myelin maps that I've shown you before, and we can take spatial gradients to inform us of where there are transitions that are informative about functional and areal subdivisions. We can take the thickness maps I showed you before and use those where they inform us about transitions in thickness. We can take the functional task activations that I've already illustrated and use each of those interestingly distinct task contrasts to get more information about functional transitions. And we can use the fMRI resting state data, bearing in mind that it's powerful but not identical to some of the cortical areas. And that's a work in progress, but I just want to illustrate one specific example of a set of cortical areas that are quite small and distinct and are revealed by this multimodal analysis that Matt has done. So we're concentrating on this little patch of two heavily myelinated patches, one of which is known to be the frontal eye fields just in front of motor cortex, and another that's suspected to be the premotor eye fields, and then a lightly myelinated patch in between. When we do the spatial gradient of myelin, we see transitions that are candidate areal boundaries, and those turn out to correlate very well with the task fMRI contrast that I've shown you before that has a sharp spatial gradient collinear with these myelin transitions. And if we take the resting state fMRI, here's a seed location in lateral temporal cortex that lights up strong functional connectivity with this same domain, whereas another seed in parietal cortex selectively avoids this middle patch and lights up functional connectivity with these two eye field domains. And then the icing on the cake, in my view, is that Matt went back to the classical literature and found that, lo and behold, in 1956, Hopf, a classical anatomist, identified a lightly myelinated strip identified as area 55B. And if that was the only thing going, you might be suspicious of it, but it actually correlates very well, and we use that as an argument that we have multimodal, a modern and classical delineation of a set of areas, including this little tiny but intriguing area 55B. So there's a long ways to go, but we're optimistic that this kind of careful, systematic, multimodal approach will inform us about a large fraction of the collection of upwards of 200 cortical areas in the human cerebral cortex. Another way of approaching this, going back to resting state fMRI alone, is what's called independent components analysis. And this has been pioneered by Steve Smith and Christian Beckman in our consortium. The basic idea is to focus on the commonalities in the resting state signal and use an objective independent components approach to identify the spatial maps in a way that is essentially a soft parcellation that tolerates non-contiguous patches or networks. And then one can use these data to identify connectivity matrices of the type I showed you for macaque cortex from ages ago, but now using the resting state data and using either full correlation or partial correlation analyses to infer the connection strength as revealed by this modality. So it's not a direct surrogate for anatomical connectivity, but it's an estimate that is valuable to have. And using this approach, Steve Smith has carried this out with many different levels of granularity or spatial components, and we've made this resource available to the community to start exploring functional organization and functional circuits using all this information. We've also provided new methods for visualization. So here's the Connectome workbench vision of cerebral cortex and subcortical structures for one component. That's the default mode network that has been intensively studied by many labs. And we can take this connectivity pattern and threshold it and identify the strongest components and do basically whatever we want. We can look at just the positive connectivity components and generate a parcellated connectome read out one row at a time. So this is the default mode network. Here from the same data set is the attention, dorsal attention network that we can explore and probe at will using this offline visualization tool. The same data sets are available online, and a visualization tool developed by the Oxford group will soon be online so that you can probe the same data interactively without having to download anything, but just point and click to the default mode network and its functional connectivity, positive and negative, viewed as a whole, or looking at that network subcomponent alone. So these tools are going to be very rich exploratory options for looking at functional connectivity and also comparing it with behavior given the richness of the data sets that I've alluded to before. So those are works in progress that are related to the multimodal and especially the resting state network data we have. I want to say a little more about the cerebellum. I think it's a special structure, but because of the challenges I mentioned before, we can't reconstruct the cerebellar sheet in every individual, but we can map it to an atlas, individual subject cerebellar atlas map that I generated some years ago, and we can view that on inflated and flat map versions and then paint it to show the different cerebellar lobules to orient you to the different subregions, and then we can probe the functional connectivity of these regions. As a starting point, Randy Buckner's lab has not only identified major resting state networks for the cerebral cortex, but they've done it for subcortical structures and for the cerebellar sheet, which we've mapped onto our surface map of the cerebellum. So we can take these 7 networks and zoom in on the default mode network that I mentioned before, a very intriguing and popular domain of analysis. So here is the Buckner Labs version of the human default mode network in medial and lateral cerebral cortex, in the caudate nucleus, and also in several major patches of the cerebellar sheet. So the default network that is active when you're at rest and suppressed when you're thinking about many different tasks involves these major domains. One of the things we found, to my surprise, is that when we take the listening to stories, which is an active task, and compare that to doing arithmetic from our Connectome project data set, the story versus math contrast lights up the entire default mode network in cerebral cortex and in the cerebellum. So it's an intriguing characterization. But when we look more closely at the default mode network, we can take the resting state data that I've touched upon before and the independent component analysis that I mentioned. We can use an intermediate range 100-dimensional ICA decomposition and see that the default mode network is no longer a discrete complete network. We can look at network components such as this lateral parietal patch, and then over in the cerebellum, the hotspots of the ICA decomposition of this network is no longer in the Buckner version of the default mode network, but is lying outside of it. So all of this is a pointer to the richness of the connectivity and functional organization that we, the community, can continue to dive into and try to understand and interpret. But the answers are going to be intriguing but challenging to carry forward. In the last few minutes, I want to touch upon the other modalities that I just haven't had time to delve into in as great detail. Tractography relies on diffusion within the white matter of the brain, which is organized essentially as cables, but with tons of crossing fibers for the reasons that I mentioned before, because the connectivity matrix that underlies human cerebral circuits must involve all sorts of crisscrossing fibers that we need to decipher. Diffusion imaging captures not only the dominant direction of or orientation of fiber bundles, but can be used with the high angular resolution that we achieve with the Connectome project to estimate fiber orientations at 2 or even 3 crossing fibers at each little voxel. And we benefit from the higher quality of the diffusion imaging data in the Connectome project. And you can see it at a glance. This is a map of fractional anisotropy that's an indicator of the higher quality of HCP data compared to 2 other standard data acquisitions. Peter Kochanoff has analyzed this fractional anisotropy, so this is not looking at pathways, but at just a measure of white matter organization in the deep white matter. And we've taken advantage of the heritability approach that our twin data sets from the Connectome project to show that the heritability of fractional anisotropy is considerably higher using the HTP data than has been seen using other approaches that don't have as high a data quality. So it's an intriguing next step in thinking about white matter organization. We can use the Connectome project tractography data to capture better representations of deep white matter tracks, and also we can represent the long distance pathways from one region of cortical gray matter to another. We can visualize the trajectories within the white matter, and we can visualize the structural connectivity in individuals and in group averages to complement the functional connectivities analyses I've mentioned before. So here, for example, is a side-by-side comparison: structural connectivity from tractography in one individual with a seed in temporal cortex, and from the same individual, here is the functional connectivity map. and you can see broad similarities and many strong correlations. That's the good news. But you can also see strong and non-trivial differences between these two modalities. Some of these may reflect artifacts that are associated with one or both modalities. That's the reality we have to deal with. But some of them may reflect the more indirect nature of functional connectivity, which does not show, is not restricted to showing just direct pathways. Just a whiz-bang fly-through to remind you that we have another modality of magnetoencephalography whose strength is that it provides millisecond level time resolution, but whose price is it's spatially much coarser. Previous studies have shown that MEG can be used in both the resting state and task activations to identify functional networks of the brain. We're acquiring from these 100 subjects resting state and 3 different task activations. We've released some of the data, and we've started with our colleagues in Chieti, Italy, to analyze some of these data, capitalizing on the access to different temporal signatures. So if one analyzes from a group average of 8 subjects as an illustration of proof of concept, a measure of functional connectivity for different network components in the alpha frequency band, a moderate frequency range, one sees functional connectivity within the visual network and between the visual network and the default network. Whereas if one takes the same subjects and analyzes at a different frequency band, just one notch higher in the beta band, it's quite a different pattern of functional connectivity. So this is just an illustration of the attractiveness of having these multiple modalities from the same subject available for exploration. And lastly, I want to circle the wagons, as it were, and come back to the interspecies comparisons. We spent a long time on human cerebral cortex, but I started with animal models, and we can make comparisons between human and monkey cortex by aligning functionally corresponding, presumably homologous domains, and we can make maps of the cortical expansion of human cerebral cortex that is vastly expanded in lateral, parietal, and temporal and frontal cortex. We published this expansion map previously, but now we can relate this to the myelin maps that I've shown you previously, which we've acquired not only for human cortex, but for monkey cortex. And this is a group average monkey myelin map, a group average human myelin map, and you can see many similarities, but what you can see is that the higher cognitive regions that have expanded greatly during human evolution correspond to the lightly myelinated patches in both humans and macaques, and lo and behold, also in chimpanzees, where Matt Glasser was able to collaboratively get access to chimpanzee data that we made myelin maps of. So this is just an indication that the importance of combining our information not only from multiple modalities, but multiple species to attack these fascinating questions of how is the human brain and the mammalian brain in general organized. It's a grand challenge for our century, if not the millennium. The Human Connectome Project is making advances that don't answer many of the critical questions, but take us a major step forward. We'll get important discoveries and insights, but we're not going to get a complete human connectome from the Human Connectome Project or any other modality currently in sight. So we have to be realistic, we have to be optimistic, but maintain a critical eye on the data sets that we're acquiring and analyzing. We are optimistic that the improved methods that I've touched upon in different modalities for acquiring, analyzing, and sharing data will generalize to other projects and will lead to improvements in studies of diseases and development and aging that will have clinical impact in the long run, we hope. So a final cartographic analogy. We've gone from classical Earth maps and classical brain maps to improvements in book atlases of brains and Earth. Satellite imagery was revolutionary for analyzing the Earth. MRI and other imaging modalities have been comparably revolutionary in the past decade or two. Google Earth has wildly transformed the way we navigate our Earth's surface, and we are hopeful that continuing improvements in visualization and analysis methods will give us deeper insights about the human connectome in health and disease. So again, I've got to give special thanks to an outstanding group of investigators. I can't name them all, but I would be remiss if I didn't highlight the tremendous contributions of my co-PI, Camille Ugerbill at Minnesota, Deanna Barch at WashU, Steve Smith at Oxford, Dan Marcus on the database front at WashU, and my exceptionally talented graduate student, Matt Glasser, and of course our funding agency that contributed to the essential effort. So thank you for your attention.
Thank you to Professor Van Essen. None of this would happen without sponsors, and we've got some fantastic sponsors. All of our sponsors are fantastic, I should say, in the Life Sciences Forum. The Bionics Institute, RMIT University, the Victorian Life Sciences Computation Initiative, the Melbourne School of Engineering, Melbourne Health, St Vincent's Hospital, Phillips Ormond Fitzpatrick, IBM, the Melbourne Convention Bureau, the Club Melbourne Ambassador Program, and the Centre for Neural Engineering, and the Australian Regenerative Medicine Institute. They make all of these events possible. It's wonderful to have so many organisations come together. They all believe in communicating science and engaging with the community. So thank you and good evening.